Hi, everyone. Welcome to SWIM. My name is Claire. I'm a senior scientist at the Good Food Institute. So just some quick background on GFI for those of you who aren't familiar with us. We're a nonprofit that seeks to accelerate innovation in alternative proteins, by which we mean cultivated meat, plant-based meat, and also fermentation-derived proteins. So this webinar series is part of the Sustainable Seafood Initiative, which is something we've been doing for the past 3 years or so, which is basically, the same idea as applied to seafood. We really see seafood as a little bit of a neglected area where we need more awesome research like what you're going to hear about today. So that's kind of the background on who we are and why we're here. So just a few housekeeping items. We are recording this webinar, so it'll be available on our YouTube channel. If you see at the bottom of your screen, there should be a chat and a Q&A box. Please use the chat to connect with other attendees, make excited comments, whatever you feel like using the chat box for. That's what it's there for. We encourage you to use that. If you do have questions for the speakers, please don't put those in the chat. Please use the Q&A box. That way it makes it much easier for us to see what's been asked, what's already been answered. And just makes it a little bit easier to keep track of everything. We have a networking session following this event. I'll put the link to that in the chat. So this is using MeetAway. It's basically a series of 8-minute video calls. You'll be connected with other people who are working on alternative seafood, and you can kind of match according to common interests. We also have a second session that's on Tuesday the 15th, and this is just for time zone reasons. So if you're not able to make the first one, or if you're watching this later on our YouTube channel and it's before Tuesday the 15th, you're welcome to check that out. If you are in the United States, it may be in the middle of the night for you. One other thing I wanted to mention is our seafood newsletter, Turning the Tide. So this is quarterly. We don't spam your inbox, I promise. Oh, and I just realized I'm sending links only to panelists, which was something I wanted to remind everyone of, is I think for attendees, it will default to sending it only to panelists. And you need to switch that if you want to talk to everyone at the event. So that first link is the networking session right after this. That's the one on Tuesday, and this is our newsletter. I also wanted to mention that GFI is supported by philanthropy. And so if anyone in the audience is part of our family of donors, we really appreciate you. Thank you so much, and we really couldn't do what we do without you. So without further ado, it looks like we have a number of attendees already here. Thank you guys so much for joining. So I will go ahead and introduce our two speakers and then let them take it away. So our first speaker is going to be Dr. Reza Ovissipour. Reza is an assistant professor at Virginia Tech, and his group is working on a project to optimize media composition for fish cell culture. We also have Lexi Dusher, his postdoc here, who will be joining in for the Q&A portion of this. Our second speaker will be Dr. Cameron Semper. Cameron's a postdoctoral fellow at the University of Calgary, and he's working on a project related to species-specific growth factors for a variety of seafood species, and really trying to kind of make sure that we're using not only growth factors that work, but the best growth factors. So with that, I will stop talking and turn it over to Reza.
Thank you so much, Claire. I would like to share my screen. Can you see my screen right now?
Yes.
Right. So great. Well, good morning, everyone. I think you are from all around the world. Good afternoon, good night. So thank you so much for attending in this webinar. I'm so excited to be here and talk about some part of our results that we have so far from this project. We are working on optimizing growth media for seafood cell lines using machine learning techniques And before jumping into the presentation, I would like to express my sincere thanks to those who have been working hard on this project, including Dr. Lexi Dusher, my students Inayat and Ted. And also I would like to express my sincere thanks to Dr. Claire Boomkamp from GFI, who has been helping us during the last 2 years. And the funding agency is GFI, and we do really appreciate the opportunity that they provided for us to work on this project. Also, I would like to express my sincere thanks to some of my collaborators from Hume Center for National Security and Technology at Virginia Tech and Commonwealth Cyber Initiative. They have been helping us with developing algorithms. Virginia Tech main campus is located on Blacksburg, in Blacksburg, and we have 11 agricultural research extension centers, and we are located in one of them, which is Virginia Seafood Agricultural Research Extension Center here in Hampton, pretty close to the ocean. And this is our new building. We are really excited about this new building. It's a 15,000 square feet building, and we are going to have access to several different pilot plants, including cell-based lab. And we are supposed to move into the new building sometimes in November 2021. Virginia Tech Seafood AREC is located in the center of the one of the largest seafood producers estates. Virginia is the third largest seafood producer in the nation, and we are collaborating with other institutions in the state of Virginia to address food safety, food security, and sustainability. Particularly, we are working very closely with seafood industry. A little bit about our unique center here. Not only we have a really interesting lab for cellular agriculture and sustainable food production systems, but also we have access to different types of aquaculture facilities. We have different types of tanks for keeping different states of the marine organisms or seafood. We have access to fresh water and salt water, warm water and cold water resources, and technically we can keep any type of seafood live at the same time in the center. We have right now pompano, marine species, warm water, and rainbow trout, cold water, fresh water at the same time in our facility. We have different types of hatchery systems, and we have been designing and producing all of these hatchery systems using our workshop. And also we have a really nice 3D printer to print the different devices that we need. And we can spawn different species and have access to different type of seafood in very unique state. We have a really nice algae room feed manufacturing, and also we have access to other seafood species through our collaborators, and we can bring technically all the seafood that we want into our facility and work with it. My lab here is a sustainable food production system lab, and I've been working on alternative proteins, bioprocessing, cultivated seafood, and AI and machine learning on algae, plant-based protein, insect-based protein, and recently we started working on the cellular agriculture or cellular aquaculture as well. So what motivated us during the last 20 years that I have been working with seafood was the problem that we are facing in terms of the food security and food sustainability. Particularly for the seafood industry, we have so many problems that this industry is facing right now, like byproducts, production, fraud, poaching, emerging diseases, superbugs, antibiotic-resistant bacteria, microplastics. In my lab, we have been trying to address some of these issues by using novel technologies, value-added products from the seafood waste, and also using some alternative proteins for feeding the fish. But technically, in a long term, in 25 years vision, we need to have access to the novel technology other than what we have been doing technically at the lab scale. So fortunately, cell-based industry is really growing at this moment. And like any other supply chain, it requires to have access to the cell bank, cost-effective media or production platform, a scaffold, and the same or proper flavor and texture, and also proper life cycle analysis. At this moment, we are working with cost-effective media, and we are trying to use different technologies for optimizing the media, because this is one of the major bottlenecks in this industry, at least for the seafood. And seafood cell lines haven't been really studied very well, and we need to develop some novel media and screen them really fast for them. We have been using different technologies for optimizing different media for yeast, bacteria, and algae in our lab. And also we were using different RSM methods or response surface methodology for bioprocessing optimization. So, but this is the first time that we are trying to apply whatever that we have been using for other microorganisms for cells, seafood cell line here. We know that technically one of the major challenges that we have is just the fish cell line have not been really studied perfectly. So let's take a look and see what we have when we are trying to use the conventional method. When we are dealing with the conventional methods of analysis or experimental design, it's one factor at the time. Technically, this is one of the simplest method or conventional method that we have been doing in the lab. It's very robust and simple, and it's widely used. But the problem with that is if we want to have access to the big data set, we need to use the high number of the experiments, and we cannot really see the interaction between the data, and then we cannot really develop the predictive models. But compared to that, when we are working with response surface methodology, technically we can reduce the number of the experiments. It can provide some variable interaction easily, and based on that, we can develop the predictive model. But what we have been learning during the last couple of years working with different other microorganisms, including eukaryotes like yeast, we found that RSM does not mimic really good the biological system. And that's why that in this proposed work, we are also working with artificial neural network, which mimics the biological systems, creates effective predictive model, and can handle large amounts of data. And also the most important thing here is by running the less experiments, we can train the artificial neural network, and it can learn from whatever that we are feeding into the system, and it can predict the next step. Instead of for RSM, for example, we need to have some set of data and experiments, and we cannot really change that. The system cannot be trained. The system can be actually fed by the number of the experiments that we are running at this moment. One of the as you can see here, so we started our experiments with RSM. So we selected 7 different growth factors that we selected them from mammalian experiments technically, and we had different range of the chemicals, as you can see, for each of them. And then we fed them to the model. It created the meat part of that for us. For example, for TGF, we had 1 to 6 nanogram per milliliter. We determined them as minus alpha and plus alpha, and then it picked this middle part of that for us. And then it designed the experiments for us, and we had 92 experiments, and the experiments is designed based on the best interaction among these 7 growth factors. So technically, this is the central composite design. After running these experiments for 6 days and replacing the media for 2 times, we found that this media, which is completely serum media, after 2 days, the survival of the cells, they were not really satisfying. So we started seeing so much deaths after 2 days. And that's why in order to develop the platform and understand whether the model is going to work with that or not, we decided to add serum to the media as well. So right now we have 8 different growth factors, including serum from 0 to 10%. But our model, we designed a model to reduce the serum application and get the best results at this moment. And then later on, we are planning to reduce the serum application and make it 0 actually in the final product, in the final media. We are working with different softwares. Not only we are optimizing the whole process, but also we are trying to see how we can use different softwares for all data sets and optimize that as well. So we have been working with the zebrafish embryonic stem cell line from ATCC, very well established cells, and then we have them on with the basal media, and we add all of these growth factors to the basal media, and at the determined concentration experimental design that we have from the RSM. So what we found so far was when we are running the RSM, technically, if we want to have the same number of the experiments with the 8 factors at 5 different levels, we only need 92 experiments with RSM compared to 160 experiments with the 1 factor at the time. And also we can easily develop the predictive model and we can adjust the optimum condition. For example, here, as you can see, we have ascorbic acid versus selenium, and it gives us the optimum, the model, and also FGF versus IGF. As you can see here, by increasing the IGF concentration, we don't see that much improvement in the growth. Technically, these are the things that we can, that software will visualize for us. But also we can play around with the components. For example, when we see the IGF, by increasing the concentration, we don't see that much growth. We can easily reduce when we are trying to order to machine or to the software to determine the optimum condition, we can reduce the IGF concentration to mid part or even to the minimum part. And always we need to actually keep the FPS at minimum level in order to optimize that based on that. And also the response needs to be kept at the maximum level, which is the growth in this case, after 4 days. So, but when we are using the artificial neural network, the system is completely different. We have the input, which are the growth factors that we are feeding into the system. And usually we have the hidden layers, mainly people, they have been working with 1 layer, but it would be better that we work with 2 layers of the hidden layers. We train the model by using these hidden layers, and then we will have our output based on that, and then we can develop our predictive models. So we did that for different days of the experiments, day 1 to day 4, and then we got very interesting results. For example, based on the data that we are feeding into the system, we can determine what variable or what growth factor has the most important factor or the value during the day 1. We found that it's called variable importance. We found that during the day 1 and day 2, FGF and TGF are the most important component. And surprisingly, same results that we got for RSM, IGF at higher concentration doesn't have a really positive impact on the growth. So technically, we don't need to apply IGF at higher concentration. So serum was not really important in day 1, and in day 2, it becomes a little bit more important. But so when we go to the day 3 and day 4, serum becomes really important. So technically, that's very matched with our first series of the RSM experiments that we started seeing some deaths after 2 days, because after day 2, serum is becoming more important for the growth of the cells. So this is the model that we can use and understand, predict the growth, develop the model, reduce the number of the experiments, screen the growth factors, and meanwhile, understand the impact of different growth factors on the growth of the cells at different stages. So for the growth rate or a specific growth rate, since we are using the 4 days, when we put that together, serum was the most important one, and then ascorbic acid. But again, for the IGF at higher concentration, it was not that much important. Technically, it tells us for the next experiment, so if we want to use IGF, we have to use lower concentrations instead of just using the 30 or 25 nanogram per milliliter. So we are working right now with 2 types of cell lines. In next step, we are going to work with the European sea bass and see what is the difference between these 2 cell lines and if we can actually optimize the media for both of them. We are trying to apply different growth factors, including but not limited to lipids, amino acids, albumin, and other components. And so since right now the model works, we can easily apply that for screening the media using RSM and ANN. We are also planning to gradually reduce the serum in the media, which from 10% to 0%. It did work perfectly for a catfish cell line, and it took 32 days to adapt to the new condition. We would like to try that and see how it can work in our model. And also we are planning to use vibrational spectroscopy for cell performance and media screening as our next step. We have a patented technology with UC Davis Technically, we had different types of biological materials, including pure DNA and protein, and different types of single cell organisms. And we had inputs into the system, and we had outputs or responses from these biological material. So by using the vibrational spectroscopy, we were able to measure and quantify the responses for the first time. So each spectra is a 2000 data point spectra. And we measured them and then after that, so we determined the fingerprints. For example, here is just the protein, nucleic acid, and lipids. And so then after processing to PCA model, each 2000 data point will be converted to one data point here. So it can actually reduce the size of the data in the 3D dimension. And we can actually have the loading plot to understand which component has the most important impact and which one has the least impact on the growth. And then based on that, we can easily develop the predictive model with a satisfying R-squared. This is another step that we are trying to apply. And we believe this is going to be very, it did work for different types of cell lines, single cells. And we believe this would actually help us to screen more media growth factors. Technically, we have the inputs and then we have responses using the conventional methods, and we measure the responses using the vibrational spectroscopy. And then based on that, we develop the predictive model, and we use the same concepts which already been proved for other cell lines or single cell for fish cell lines. In summary, what we have been observing and learning was optimization is a complicated process. It's not really easy process, and we need to apply different mathematical modeling, and it requires more robust mathematical modeling like ANN in this case. Less known about fish cell line compared to mammalian cell line, which makes it really hard. We have to adopt whatever that we have in mammalian cell line for the fish cell line, but technically they are different and they have different metabolism pathway, and we have to do more screening at this moment. For each cell line, probably we need to have new optimization, and we will actually see that very soon after running some experiments with our CBAS. Both of the RSM and ANN, they help us to reduce the number of the required experiments. And also we could actually optimize we can change the optimum conditions by manipulating the data that we have, the range. Actually, we can give different range to the model, and it can give us different optimum condition based on whatever that we feed into the model. Overall, ANN provided more robust model compared to RSM with a better prediction model and fitting ability, and more experiments with less to no FPS is required. And we are working on that at this moment. So we have different hypotheses and plans for near experiments for future experiments. And also we found out that utilization of different components are different in fish cells, and that's why that we are not really seeing the similar results that other people that they are seeing for the mammalian cells. And technically, it shows that for seafood cell lines, we need to focus more on more robust system for screening more growth factors. And if you have any question, we will be more than happy to provide after the meeting. And thank you so much for your time.
Thank you so much, Reza. That was a great talk. Really exciting to see what your lab has been up to. Yeah, so if anyone has questions, please pop those in the Q&A, and we will get to them after Cameron's talk. And so with that, let's turn it over to Cameron.
All right, so just bear with me here. I'll get the screen share going. All right, can you see the slides okay?
Yes.
Okay, great. Okay, yeah, thanks so much. My name is Cameron, and thanks to everybody in attendance today. Excited to talk about some of my research, which started with a question, and then we had a problem along the way, and this research is talking about really addressing that problem. Before I begin kind of in earnest, I figure there's people here from kind of all around the world, so I'll just give a little introduction to where this research is being conducted. So I'm located at the University of Calgary, which is located in Calgary, Canada. It's the city shown at the top here. It's close to the Rocky Mountains. It's very nice this time of year. It's very cold in the winter, but to be fair, it's a pretty accurate description of most of Canada. So Calgary is in the province of Alberta, which is shown in red here. And you'll notice that this is a landlocked province. And I get a lot of questions related to my research about, how did somebody in a landlocked province with no real fishing industry get interested in cultured seafood? And there are many reasons, but a kind of fun one that I like to give is that when people from this province and I'm sure many other landlocked states or provinces, when they vacation to the coast, something that they're often very excited to do is get kind of fresh fish, right? So stuff that hasn't been frozen, hasn't been transported long distances via refrigeration or whatever. And what's really exciting to me about the potential of cultured seafood is that the production facilities can theoretically be located anywhere, so you can get fresh fish in landlocked provinces or states or countries or whatever. So okay. On to the actual research. So today I'm going to start with an exercise. So humor me here. We'll go shopping for fish cell lines, right? So this is one that many people affiliated with GFI will recognize because it's been made commercially available thanks to their efforts. So it's a European sea bass line. Our previous talk just mentioned they plan to begin working with it. So that's great to see it now available. So let's say that we want to buy this cell line. We want to get ready for its arrival. So we want to look at all the things that we need to order to support these cells and their growth. So we look at the growth conditions and you see, okay, it requires L15, which is a type of basal media, and 10% fetal calf serum. All right, so we'll order that one, but maybe we want to get more than one fish cell line. So we look at another one. This one's from a species called bluegill, which is common amongst recreational fishermen in North America. So what do we need to grow this cell? So we need EMEM, some other supplements, and again, we need 10% fetal bovine serum. Okay, one more time, zebrafish line. This is a different zebrafish line than our previous talk discussed, but this is a model organism. Should be more widely used in research than perhaps the previous cell lines. So what do we need for this one? And looks like we need DMM F12, and again 10% FBS. So we could go on and on through pretty much all the fish cell lines. I'll spare you all that. But here's a list of a selection of cell lines that are either available commercially or that have been reported in literature. And you'll notice that all of them are using between 10 and 30% fetal bovine serum. So this observation was pretty counterintuitive to me when I noticed it. If we think of a cow, which is obviously the fetal calves, which is where the bovine serum is coming from. They're terrestrial, they're endotherms, large mammals, they live on land. If you think about a fish, it's an aquatic organism, it's an ectotherm. The environment that these organisms develop in is drastically different. One's developing in kind of a more constant, highly regulated temperature environment, the other's developing in water, which can change in temperature quite drastically. These organisms diverged, more than 350 million years ago. So there's been a lot of time from an evolutionary standpoint for divergence of various, cellular pathways. And this led to the question of, is FBS really the best supplement for supporting fish cell culture? So that was a question that I had and one that I wanted to go about looking into. So just a quick primer on FBS. So it was mentioned in the previous talk. I'm sure many people are familiar with it. So it's a rich supplement used to support primarily mammalian cell cultures, but as we've learned from our previous slides, also fish cell culture. It contains hormones, vitamins, growth factors. For my talk, growth factors refer to the small protein growth factors that I'll be talking about as we proceed. Lipids, trace elements. But there's a number of issues with FBS. So it's costly. It suffers from batch to batch variation that can correlate with seasonality or even geography. And obviously it's harvested from fetal calves. So one of the major promises of kind of cultured meat is its potential to reduce animal suffering, but reliance on FBS sort of negates that advantage. And with that in mind, and as our previous talk said, there's really a commitment within the field, both from the industry standpoint as well as academic research to, eliminate the use of serum. It's not ideal for growing cells, and there's the other concerns as well. So there seems to be a consensus to work towards its replacement. So replacements or alternatives exist. This is called serum-free media. It's been around for decades, and it replaces the bovine serum with defined components. And commonly, it relies on recombinantly produced growth factors for the replacement of serum. So there's a plethora of different serum-free formulations out there. Some are specific for particular cell types. And while I said FBS is expensive, turns out serum-free media can actually be even more expensive. So this is a recent cost analysis was done last year by GFI, and what they found is when they analyzed a common serum-free formulation called Essential 8, they found that growth factors or protein growth factors are major cost drivers. So this analysis here showed that really more than 95% of the total cost can be attributed to these two protein growth factors, FGF-2 and TGF-beta. That's pretty wild. But as somebody who has a background in protein biochemistry and recombinant protein production, this also looks like potentially a significant opportunity for facilitating cost reductions. Okay, so these growth factors, right? Why are they so expensive, right? You saw there for that scaling cost analysis. There were millions of dollars. So. They're difficult to produce, right? That's the simple explanation for why they're so expensive. So some of them are produced recombinantly using microbes, but this is usually plagued by low expression and insoluble protein that forms in what's called inclusion bodies, which then must be purified under denaturing conditions and subsequently refolded for it to regain its bioactive conformation. So this adds time and expense to the process. In other cases, they're actually produced recombinantly using eukaryotic cell culture, and that just is associated with significantly higher production costs than microbial production. But in some cases, that's what's required. So taking all this into account, this kind of led to the project that I'm working on now, which is basically I'm looking and seeing what sort of fish growth factor orthologs are out there, and is there sequence diversity compared to mammalian growth factors? And then I have to, so it turns out you can't really buy fish growth factors, at least not off the shelf. Maybe a custom order, you could get some, but it's a difficult thing to buy. So we had to work out producing them ourselves. That's the next phase of my project, which I'll be talking about mostly today. And then we'll be using those growth factors to create serum-free media that's optimized for fish cell cultures. So back to my initial question, which is, this idea about using bovine serum for fish cells. So to me, it seemed that there are really two possibilities. So one is that, there's such a high level of conservation in these cell signaling pathways that are, activated by these protein growth factors, that maybe FBS is actually really well suited for fish cell culture. The other possibility is that, maybe the use of FBS is more because of a history of its use in cell culture, precedent inertia, resistance to change, and that there are better options. And potentially the fish growth factors may represent one of those. So to analyze this, the first thing I did was I identified fish orthologs of mammalian growth factors that are commonly used in cell culture, and that will likely play a role in cultured meat applications. So here you see that we compare the fish growth factor sequences from a selection of fish species to their mammalian ortholog, and you see a bunch of percentages here. This is the shared amino acid sequence identity, which may not mean a lot to everybody in the audience, but to a protein biochemist, this level of sequence diversity is actually where we start to see real differences in protein behavior. So just as that's a theoretical example, two proteins that share, say 75% sequence identity, they may be performing the same role biologically, but one might be much easier to produce recombinantly than the other. Maybe it's more stable. Perhaps it's resistant to proteolysis compared to the other one, or maybe it has higher potency. And I come from a lab that works primarily in structural biology, and we actually exploit this level of sequence diversity routinely to find protein orthologs that will crystallize and allow for structural characterization. So, I've had examples where, I have two proteins that are very likely performing an identical function biologically. They share, 85% sequence identity. One protein is really challenging to work with, and the other one, crystallized readily and gave me a beautiful three-dimensional structure. So seeing this level of sequence diversity was quite exciting, and it kind of told us that it was probably worth seeing, what we could discover in terms about the ease of production about these fish growth factors, as well as, what sort of bioactivity they may have. So just going on to the specific growth factors. So our previous talk mentioned some of these, and I'll go into a little more detail about them. So the first one is called insulin-like growth factor 1. It's a small disulfide protein that is structurally similar to insulin. So it's found in fetal bovine serum. So it's one of the things being supplied by that supplement. And it's also used as a component kind of interchangeably with insulin in many serum-free formulations, including in some compositions of essential 8. So it binds to its cognate receptor called the IGF-1 receptor. It can also bind the insulin receptor, and it stimulates cell growth and proliferation through the PI3K and ERK pathways. So you can purchase mammalian IGF-1 commercially. It's produced microbially and purified from inclusion bodies. But at least I couldn't find fish IGF-1 for purchase. So the question that I posed previously about whether or not these will improve the performance of fish cell culture required me to solve this problem first, which is purifying them in a reliable way. So we wanted to see if we could produce these fish IGF-1 sequences. And to do that, we tested a number of different expression vectors and fusion partners. So on the left here, you can see our first attempt. So this was with a standard fusion partner. And we were actually just hoping that we would get lucky and that perhaps the fish IGF sequence would be more amenable to purification under these conditions. But unfortunately, we didn't get any expression. So next, we tried another fusion partner. You can see that the protein here is denoted by this symbol here. And we saw that the protein was expressed, which is shown in this lane, but the soluble protein, which shows up here in this S lane, wasn't present there. So that tells us that the protein is not properly folded, likely formed inclusion bodies. So this is kind of the status quo situation with what's available currently. So then we tried some pretty unique fusion partners that we repurposed for growth factor production. And you can see here that we've had a pretty dramatic improvement. So we get a nice level of expression, and it turns out that protein is actually soluble. So we're able to purify it without any sort of denaturants or anything like that. So it's a significant improvement. And then we tried another one, which is shown on the right here. And you can see here that this improved things even further to the point where we're getting really true overexpression, and we're retaining a significant portion of that protein as soluble folded protein. So these are kind of expression experiments. Turns out that we can actually purify these readily. So here's some purified samples right here. These are fish IGF-1 orthologs that purify readily. They're pretty easy proteins to work with when they're associated with this fusion partner. So this was pretty exciting. So if we look at our purification results, so we didn't do much optimization beyond the vector design and the fusion partner identification. Here are our yields here. And if we look at, I did some shopping in the last couple of days, commercially available IGF-1, it's not that expensive. I found it ranged from about $350 to $800 Canadian per milligram. I did a quick calculation in terms of our expenses. The caveat being, well, I did include the cost of labor. I did not include the equipment that we used, which would be, like upfront capital costs. So please take that into account. But our costs of using this methodology range from about, $11 to $40 per milligram. So, quite a bit cheaper. So that's great. Cheaper is great, but if it's not active, it doesn't matter. So thanks to my colleague Mina at the University of Toronto, we were able to screen these recombinant IGF-1 orthologs for biological activity on 3T3 cells. And what we found is that IGF-1 produced using our methodology is comparable to commercial recombinant mammalian IGF-1. So here you have the mammalian in red, and these are fish orthologs shown in green, purple, and blue. And you can see that they're giving a comparable level of performance to the mammalian IGF-1. So on to the next one. So this is a really expensive one, transforming growth factor beta. So it's found in FBS as well as many serum-free formulations. So it's one of the most expensive components of those media formulations. It's typically produced actually using eukaryotic cell culture, and that's one of the reasons why it's so expensive. So acting in concert with FGF-2, excuse me, it supports undifferentiated cells, and it binds to its cognate receptor and activates the SMAD signaling pathway via phosphorylation event. So our initial approach is very similar to the approach we took with IGF-1. So we tried a very simple fusion partner, again, trying to exploit that sequence diversity associated with fish TGF-beta. Surprisingly, we actually got a bit of expression here, but no soluble protein. So, you could pursue purification of this from a denaturing protocol and refolding, excuse me, but we tried something else. So we tried a second fusion partner. So we see, expression has improved a little bit, but no soluble protein. Tried our next fusion partner. Again, expression improves, and here we do see some soluble protein. So this is expressed, but the solubility is fairly low. And our final fusion partner, again, sorry, not the best image. It's a little dark, but you can see here this symbol denotes where the protein is, and you can hopefully see it's a kind of thick band there. So we see protein expression, and we actually see a good level of protein solubility as well. And we can purify that as well. So again, doing our little cost analysis, our yields here. So we look at the results of our purification. Again, I did shopping last night for some commercially available TGF-beta. This one's really expensive. I found prices ranging from 8,500 to just under 20,000 Canadian per milligram. I did the same calculation I did with IGF-1, and our cost to purify about a milligram of this protein is ranging from $15 to $70. So it's quite a bit less. So again, we tested TGF-beta for bioactivity. It's a little more difficult to assay than some of the other growth factors, but you can see here that our recombinant fish growth factors are active. And again, this is on a mammalian cell line. So that gripe I had about the cross-species reactivity and using FBS for fish cells could hold true with this assay as well. And we see that, although the commercial TGF-beta did outperform our recombinant one, it was, maybe by a factor of, 1.5 at most. And when you consider the difference in cost, I don't think that level of performance difference is enough to justify the expense of the commercial enzyme. And though I don't have the image here, we did show through Western blot, we confirmed activation of the SMAD through the phosphorylation. So this is exciting. And then I'll go on to our next growth factor here, which is another major cost driver. This is the basic fibroblast growth factor. It's again a major component of FBS and most serum-free media formulations. It binds to the FGF receptor. Our previous talk showed how important a component this can be, and it has really broad mitogenic activity. So I won't go into the gels in order to save time because I'm running out of time here, but I'll look at the purification here. So you can see we had really nice yields of fish FGF2s. Again, a cost analysis commercially available ranges from about $1,100 to just over $2,500 Canadian per milligram. Using our methodology, we're getting it for about $10 per milligram, so significantly less than what you'll pay commercially. And there's been papers recently from the Burridge lab out of Northwestern that have shown that they can produce the recombinant FGF for much lower cost than what is available commercially. So when we tested the fish FGF2 for bioactivity, we saw some really promising results. So again, this was tested on mammalian 3T3 fibroblasts, and our results actually suggest that fish FGF2 may have higher activity compared to commercially available FGF2. So our commercial one is shown in red here, and some of the fish orthologs that we tested are shown in purple here, and you can see that at multiple concentrations of FGF2, they resulted in significantly higher levels of cell proliferation. So this is exciting and suggests that we may be able to exploit that cross-species reactivity in the opposite direction. So while as FBS has helped with establishing fish cell lines and supporting fish cell culture to date, maybe in the future we'll be using the fish orthologs to reduce costs and improve performance in cultured meat production going forward. I'm running out of time. I don't have much time left. I'll say that we've started our cell work optimization here. We've done some basal media optimization, looking at the effect of reduced serum in different basal medias. We're showing here that, in L15, we had pretty poor results, but in NEM with our cell line, we're able to get still, robust cell growth in reduced serum conditions. So this is exciting. We need to get the serum lower, and then we'll start testing our fish growth factors. And, I'll be giving a talk at an upcoming conference in August where I'll be focusing more on this aspect of the project. So with that, I'd just like to acknowledge some of my collaborators, my supervisor, Dr. Suchenko, for letting me work on this project, even though he's a structural biologist and probably 4 or 5 years ago had no thought that he would ever work in cultured meat research. And then 2 of my colleagues in the Suchenko Lab Toronto edition, Mina and Peter, they've been a huge part of the project, and Mina especially for the bioactivity screening, which helps me know that my recombinant growth factors aren't completely useless. And then Luke, who's a research technician in our lab in Calgary, who helps me with the cell culture work. And then I'd really like to thank my funders, New Harvest and the Animal Advocacy Research Fund, both for funding the research and in the case of New Harvest for building a really great community of collaboration, and for the other New Harvest Fellows who have helped with a lot of my research as well. Thank you for your time. I'm a little over, I apologize. If you have any questions, here's some contact info for me, and I'll monitor the Q&A for the rest of the session as well. Thanks so much.
Thanks, Cameron. That was a great talk, and yeah, always exciting to see those sort of cost comparisons between the commercial growth factors versus the ones produced in just in someone's lab. I think it's just really it shows that there's a lot of potential for further optimization of those costs. So if anyone has further questions, please do put them in the Q&A. I'm going to jump into the first question. So Maynard asks, what kinds of environmental costs can be measured for such research? How do you identify, measure, and discuss the externalities of both current R&D as well as future production as you ramp up to scale? And so I'll just quickly plug the life cycle and techno-economic assessment that GFI recently commissioned. I'll put the link to that in the chat. But I don't know if maybe Reza or Lexi, if you'd like to discuss a little bit about that.
Sure. So I mentioned the life cycle analysis in one of my slides, and I think this is, I can jump in, and Lexi, if you have anything else, please feel free to add. That's one of our concerns as well, or one of the topic area that we would like to focus on. That is still, there are a couple of papers available on seafood specifically, and also the work from GFI that you already mentioned. But we did a calculation, and for example, for one of the seafood products, if we can produce them under the lab condition, the cell-based seafood, we can reduce the water application or water footprint by 99%. And these are based on the actual data that I was collecting during the last 20 years. I put everything together, and then we came up with the 99% only for water footprint.
Thanks, Reza. Next question is from Katie for Cameron. Can you explain more about the process of finding a fusion partner for each growth factor, and how you decided which to test for improved expression and solubility?
Sure. Yeah. So anyone from a protein production background will know that there's a lot of different fusion partners out there that are regarded as potent enhancers of solubility and this sort of thing. So we did try we did try some of the standard ones that you would default to, like say maltose binding protein is a really common enhancer solubility. So we tried some of those, sort of the low-hanging fruit, but we didn't get much results. So then what we did is we kind of went to the literature and searched for solutions to the problem that we thought was influencing why these growth factors aren't folding. And then we found some candidates, and then we screened a selection of them and got some promising results. So I know that's not a great answer, other than to say that we'll have a paper hopefully coming out, or at least submitted later this summer, which will give you more details about that. But also just kind of having experience in the field, knowing, having worked with lots of different proteins, that helped as well. So, but the place to start, I would say, if you have a challenging protein that you're working with, would be with the standard kind of classical enhancers of solubility.
Thanks, Cameron. Can't wait to read that paper when it comes out. We have a question for Reza. Do you think it's a good idea to compare the effects of your media formulations with other well-established mammalian cell lines or cell lines relevant to cultivated meat? And do you think this might give you critical insight?
Yes, absolutely. This is a great actually point. And after developing the baseline with the RSM and ANN, and we found out that the models, they can work. So we are planning to use different growth factors, including those that they already been established. And that's in our plan of a study.
And then I also just wanted to point out that the initial growth factors that we chose were optimized for actually mammalian cells useful for cultivated meat, like the bovine, like the B8 serum for bovine cells. So that's actually where we started to attempt to figure out what was important for our fish cells, but we found out fairly quickly that we're going to need something different in fish cells. So we can test them again to other mammalian cell lines, but we believe that we're probably going to have differences between fish and mammalian cells with the optimized serum-free medium, just based on our initial results of starting with a mammalian specific components.
Thanks, Lexi. Thanks, Reza. Next question is from Doug for Cameron. Why is the solubility and rather than just the yield significant for the growth factors?
Yeah, so it's important because it's much easier to purify a soluble protein than an insoluble protein. So an insoluble protein requires additional steps. It requires the use of denaturants, so pretty harsh chemicals like 8 molar urea or like 6 molar guanidine hydrochloride. I'm not sure if those are like food safe. I would think probably not. So you'd have to make sure that those are completely removed if you want to use your growth factors for food production. And then so you can purify them under denaturing conditions, but then you have to ensure that they're refolded properly for them to be biologically active. So under denaturing conditions, they lose that three-dimensional structure that gives proteins their function. And then you need to hope that the protein will fold into its active conformation upon removal of those denaturants for it to be biologically active. So some proteins actually refold really nicely, and people actually prefer purifying under denaturing conditions. Obviously, many of the commercially sold growth factors are purified under denaturing conditions, and they test them for bioactivity. So refolding is a possibility, but it adds additional steps. That takes time, takes expertise, so it increases your costs. Getting soluble protein means you can purify it much easier for lower cost. So that's why it's really exciting for us.
Thanks. We have a question from Greg for Reza. He's wondering if you have approximate comparative information on the growth rates or doubling times of the zebrafish cell line versus the sea bass line.
I can answer.
Yeah, go ahead. I just wanted to ask.
Yeah, you're probably going to say the same thing, but not yet is the full answer. We've been really focused on the zebrafish establishment first, but that will be our next step. And eventually, soon, we're going to work with them side by side, but we don't have that yet. So great question. We hope to provide that soon.
Thanks. Next question, I think could go to anybody. This is from Molly, and she asks, it seems like both talks focused on adherent monolayers of fish cells. And do you expect that the efficiency of the media would change once you move from two-dimensional to three-dimensional conditions?
So that's a great point. That's why what we are trying to do when we determine the optimum condition, we have a mini reactor in our lab, and we are going to try that on the 3D environment and see whether we're going to get the same results or not. But that's at the end of our experiments, technically. That's a proof of concept at the end of our experiments. But definitely that's one thing that we would like to try that under the 3D environments. Yes.
Great.
I would just add for my research, I mean, we're hoping that, you know, the cost reduction that we can, you know, hopefully pass on for media development will be applicable to 3D cell culture as well. You know, many of the growth factors that are required for the 2D monolayer are likely to still be important for 3D cell culture. So any cost reduction that we can facilitate in that respect should help that.
We have another question from Maynard, who's wondering about whether you're anxious about potential toxicity of eating cultivated seafood products. So any thoughts on concerns related to food safety or what we can do to mitigate these? Reza, I know you have a background in food safety, so you probably have some thoughts on this.
Sure. Well, so first of all, seafood, they are considered as allergen. So unless we do something with the protein structure in the seafood and using probably CRISPR to reduce that, that's one of the concepts that people they are talking about that. But regarding the toxicity, Cameron also mentioned about some of the growth factors might not, I mean, they are not toxic, but might not have FDA approval at this moment. So technically, if we shift to non-animal or plant-based growth factors or recombinant growth factors, at this moment we are not really concerned. There is an argument, or there is a kind of meeting right now with FDA and USDA discussing the details. No one knows there's no regulation. No one knows that at this moment.
Great, thanks, Reza. We have another question for Cameron from Sean. Were all of the recombinant proteins you created done in bacterial cells, and what gene delivery method did you use to insert the genes for the proteins into the cells?
Yep, we're using bacterial system. They're expressed on an extra chromosomal plasmid, so there's really no special tricks to gene delivery. It's just a standard classical molecular biology, so nothing special to report there.
Great. I have another question for you, Cameron, who's from Maynard, who's wondering about the fact that that you're based in Calgary, and he knows that there's a lot of growers of pulses and beans in Alberta, Saskatchewan, and Manitoba, and just wondering about how you prioritize cell ag products for seafood relative to plant-based products, and sort of how do you see the intersection between plant-based and cultivated seafood products, particularly from an Alberta perspective?
Yeah, I mean, excuse me, from a research standpoint, I prioritize this because I identified what I thought was a problem that my skill set could help to address. You know, that was why I started researching on that. In terms of the intersection, I know that there's a lot of excitement about using kind of pulse crops as sort of the feedstock, you know, for maybe contribution to some of like the basal media components, you know, carbon source, this kind of thing. Or even just using the pulses for the feedstock that we use to grow the bacteria that we use to produce the growth factors. So there's a lot of potential there. And then, you know, kind of personally, I think that there's a lot of potential for, you know, hybrid products. So, you know, we kind of have a separation currently about, you know, the plant-based products and the cell-based products. I think that, you know, someone who's really creative might come up with a solution that marries the two of them, and maybe, and then, you know, I shouldn't say that there are people working on that, right? Plant-based scaffolds and this sort of thing. And, you know, so I think that the two fields will start to intersect more and more, and maybe you'll find that the product that hits shelves that's really successful because it has a competitive price point uses, you know, muscle tissue from cell-based, but maybe the lipids are coming from plants or something like that. So, you know, I think that there's a lot of potential for intersection. You know, so hopefully the pulse industry in the prairies of Canada are keeping their eye on this field.
Thanks, Cameron. We're about out of time. Just one more, what I think is probably a very quick question from Sean. For Reza and Lexi, have you investigated American lobster or other crustaceans for their growth with Essential 8 with or without serum, or would you like to?
Not yet, but we are definitely interested someday in invertebrate cell culture as well. So we hope to keep in touch about that.
Thanks. So that's all of the questions and all of the time. Thank you for asking exactly the right number of questions so that we ended on time. Really appreciate that. So thank you again to all of our speakers, and thank you to all of you for attending. If you're planning on joining the networking session, we'll see you over there in about 5 minutes. And otherwise, enjoy the rest of your day. Thanks so much for attending. Bye.
Thank you.